Now it's time to take a look at animal viruses and how they invade animal cells and use the host cell to make new virus particles. So the first step to any virus being able to enter a cell is adsorption. That's where that chemical attraction between the virus and the receptor sites that are found on the host cell allow the virus to attach to the cell. If it's a naked virus, the naked virus has attachment sites on its surface that then bind to corresponding sites on the host membrane. So here we see these attachment sites, and here we see a membrane protein that locks onto that attachment site. If it's an enveloped virus, then there are spikes on the membrane that can recognize and bind to the protein receptor sites that we see on the host membrane. So one way or another, proteins on the virus find and bind to proteins on the cell membrane. We saw in bacteriophages that the bacteriophage has a mechanism for injecting then its DNA into the cell once it adsorbs to the cell, the host cell. We don't see that in animal cells, and so the method of entry depends on whether it's an enveloped or naked virus. The naked viruses, once they bind, once they adsorb, then are drawn into the cell via endocytosis. So the membrane kind of folds around the cell folds around the virus, and the virus is brought inside of a vesicle. If it is an enveloped virus, then the way that enveloped viruses work, there's two plasma membranes. So there's an envelope around. I'm drawing that big smeary, smoochy thing. That's the virus inside. When two plasma membranes bump against each other, the membranes fuse, and now the virus is inside the cell. So that's a characteristic of plasma membranes, and they're kind of the phospholipids that are in the plasma membranes. Once we get the virus into the cell, then enzymes, either host enzymes or virus enzymes, uncoat or rip away the capsid around the virus, and now the DNA, the genome of the virus, is free in the cytoplasm. Okay, so now I'm going to take a moment to remind you a little bit about how proteins are made. This is not in your slide presentation. I just thought of this at the last minute, so I made it really quick. DNA, recall, in order to make proteins, the instructions are found in DNA. But the DNA can't leave the nucleus, and the ribosomes are out there in the cytoplasm. So rather than letting the DNA out, instead we make a copy of the DNA. So we kind of break apart the two strands of DNA. So purple here, that's DNA. So we break those apart, and we make a copy of the gene that codes for the protein of mRNA. And we make it by pairing any adenine to uracil, thymine to adenine, cytosine to guanine, and guanine to cytosine. So we can make a copy of the DNA, but we make it using the language of RNA. So for instance, here we see a strand of DNA, ACGAT is its sequence. We pair the A with a U, the C with a G, the G with a C, and the A with a U, and the T with an A. And in that pairing, then, we build this string of messenger RNA that is UGCUA. And then we break this little kind of binding together, and we have produced this strand, then, of mRNA that can spool out of the nucleus, find itself a ribosome, and then the ribosome reads the sequence of the mRNA, and it assembles this string of amino acids then based on the sequence that it read in the mRNA, which of course was built from the DNA back in the nucleus. Okay, so we're going to see in a minute that DNA and RNA viruses, they have slightly different ways of doing this work. So if it's a DNA virus, and we're going to synthesize then new virus particles, the DNA will make it all the way, once it penetrates and uncoats, it makes it into the nucleus of the host cell. And there in the nucleus, it gets synthesized or replicated. So we get a whole bunch of pieces of the viral DNA right there, bunches of those pieces. We send out mRNA, we build new capsid proteins and send those back into the nucleus. And then the capsid assembles around all of these DNA bits, and we build a bunch of assembled virus particles, virions, in the nucleus, and then eventually release them from the cell. Now, there's some exceptions, of course, always to this. Pox viruses, we do all that assembly out in the cytoplasm. And if it's a single-stranded DNA virus, then before we can do any of this, we have to build a second strand that's complementary to the original strand. And so we make double-stranded DNA out of our single strand of DNA before any synthesis can occur. Okay, so if it's a DNA virus, it's fairly direct, pretty easy to understand. But if it's an RNA virus, then we have to know what kind of RNA is in there. Plus sense RNA is RNA that acts as if it were messenger RNA. So it pretends that it's messenger RNA. So if a plus sense virus particle enters a cell, then that RNA can be used immediately to start building proteins that will be like the capsid or do whatever function inside of the host cell that it needs to do. But we can't make new plus sense RNA from other plus sense RNA directly. So let's imagine we have a plus sense RNA that is U, A, C, A, G. That's our plus sense RNA. To make new plus sense RNA, we first have to make a minus sense RNA from it. So we pair up U to A to T, C to G, A to U. That was silly of me because there is no T in RNA. A to U again, and G to C. So now we have a minus sense RNA that we built based off of our plus sense RNA. Now we break this bond here so that it's a strand all by itself. And using our minus sense RNA, then we can make new, just by pairing it up, new plus sense RNA. Break this apart. There's our plus sense RNA that we can stuff inside a capsid. And so we can use that minus sense RNA over and over and over again to make new plus sense RNA. So that's what we see here. Plus sense RNA is used for direct translation, first to make the enzyme and capsid proteins, so we get all of those building up in the cell. Then we make this minus sense copy of RNA, and that minus sense RNA is used then to make lots and lots of plus sense RNA. We assemble the capsids so that it can then be, assemble the virion particles. Now with minus sense RNA, it's a little bit different. The first thing we have to do with minus sense RNA is to build DNA from it. So there's an enzyme called reverse transcriptase that's used to make DNA from RNA. So once we have this double-stranded DNA, we stick it into the chromosome of the host, and then one of those strands will serve as the template for making new viral DNA and for synthesizing the viral proteins. And so once we have a bunch of the virus proteins, then we can assemble those around the virus RNA, and we've built new virus particles. So the bottom line is that plus sense RNA, or that minus sense RNA, has to be used first to make plus sense RNA that can then be translated into proteins and can be stuffed inside the capsids that we produce. And we do it by backing all the way up to making DNA first, and then we build plus sense RNA from the DNA. Okay. Hopefully we're good there. Okay, so the last step then, well, second to last step is synthesizing. We got one last possibility, and that is with a group of viruses called the retroviruses. The HIV, so the virus that causes AIDS, is an example of a retrovirus. So retroviruses have 2 copies of plus sense RNA in them, and they use that plus sense RNA and that enzyme reverse transcriptase to make single-stranded double DNA. The single-stranded DNA is used to make double-stranded DNA. That double-stranded DNA is transported to the nucleus, where we insert it as a piece of regular old DNA, but it's viral in origin, into the genome as a provirus, and then that provirus is read, just like we saw with the steps for the minus sense RNA. So essentially, retroviruses, although they don't have minus sense RNA in them, they do almost exactly the same steps as our minus sense RNA viruses do. So once that provirus is in the DNA of the cell, the host cell, then we make new plus sense RNA from it and package that all up into a variant as we also assemble the capsid proteins. Okay, so that's the retroviruses. So we've seen DNA viruses, plus and minus sense RNA viruses, and retroviruses. Okay, so now maturation, the second to last stage. We assemble all of these RNA or DNA viruses. We assemble them. They're either in the nucleus, if it's a DNA virus, in the cytoplasm, if it's an RNA virus, and then if it is an enveloped virion, so if it's a virus that has an envelope around it, then the virus has to pass through some sort of host cell membrane. And it could be the nuclear membrane, it could be endoplasmic reticulum, it could be the Golgi apparatus, or it could be as it exits the cell, as it leaves through the cell membrane. But our virus, here's the virus inside of a capsid. Here is the DNA that's inside of it. That virus has to pass through a membrane. As it passes through, it gets an envelope. And so at the very end, we have a virus inside a capsid that then has a surrounding envelope to it. So one way or another, it has to pass through some membrane associated with the host cell to get that envelope. And then the new virions can be released in one of 2 ways from the cell. Either by shedding, that's when the virions bud through the host cell. This happens in a very controlled way, and it doesn't kill the cell. So the cell just kind of makes, over time, it just makes viruses a few at a time, and then they're shed away from the cell. Or if it's a virus that destroys the cell through lysis, then that host cell will fill up with the virus particles that have been made, and then bursts open, and the virus particles are released. And that lysis is what then causes the symptoms that we associate with the disease as the cells begin to die. Okay, one last thing to say about animal viruses. They can become latent. So a latent virus is, and this is what we see happening with herpes simplex viruses, this latency. So a provirus is built and is inserted into the DNA of the cell. And if the provirus there is at skin cells, then that's where we see the development of cold sores, fever blisters, and the sores that we associate with genital herpes as the viruses go through a lytic phase. If it's a chickenpox virus, then that provirus will be maintained in neuron cells. So in the cells that are in the neurons, usually up close to the surface of the skin. And later on in life, after having had chickenpox probably as a child, then later on in life, under stress, immunological stress, or sometimes other sicknesses will trigger shingles, then that virus will become active, and we see the symptoms of shingles. And here we can see a person who has shingles across his chest as the viruses become lytic and start killing cells, breaking cells open. So it requires, though, some cue for the virus to be reactivated. And as I said, it could be having a cold or a fever or stress or immunosuppression of some sort, and it can start that lytic cycle. And the cue may never come, and that infected person may never, ever exhibit the symptoms of reactivation of the virus.